next program that we are going to discuss is for multi-omics for infectious diseases. So here we will speak about, in this program, we will discuss about the emergence and development of infectious disease research area and how bioinformatics is contributing to this infectious disease research. So we will actually use again the next generation sequencing data and how they can be used and how they can be employed to characterize and understand the infectious diseases, the pathogens from a data perspective. So yeah. So in this program, we will show you examples of how to perform, or we will provide you training on how to perform sequence variation analysis and alignment analysis and how to perform phylogenetic analysis so that you can understand, for example, where your SARS variant or SARS strain relate to or lie in the lineage of variants of concern or variants of least concern. And we will also extend this analysis to understand the functionality and the 3D structure of the protein and how a protein structure can be related to its function and why it is important in the connotation of pathogen in infectious diseases and pathogens entry into the host. So in the end, you will also get the host side of omics data analysis. We can record what the host, how the host respond to different pathogenic infection, and from understanding the transcriptomic landscape of host response, we can get an idea of what are the different ways the hosts we as a host are trying to counterattack the viral outbreak and virus output. Will the multi-omics data teach us to make hierarchical clustering based on DNA sequence analysis for a particular gene or using? Yes, you can perform clustering using particular gene or a set of genes using coding. Yes, that is included in this as well, in this part as well. So, and yeah, okay, perfect. Let me. Can somebody mute your screen? Mute yourself. I think we have some disturbances coming up. So, okay, thank you. So yeah. So I wanted to talk to you a little bit more about infectious diseases, how it is different from non-infectious diseases, and why we are to study the differences between infectious diseases. I will not go over much detail, but we say that infections are caused by agents that we call as pathogens, and these are usually virus, viroids, fungi, parasites, and bacteria. And the non-infectious diseases are caused by nutrition deficiency or caused by chronic development of different chronic response to different factors like environment, lifestyle, and it can also come from genetic inheritance and etc. etc. So I want to give you a special overview of how omics data is actually helping in understanding the infectious diseases case. So at the same time, when we are developing a lot of new techniques to study these pathogens under microscope, we also have started developing sequencing technologies. So these two technologies, especially in infectious disease biology, complement each other. So so the data that we get and the biology that we study is rich, and it has completely transformed our understanding of infectious diseases. I don't have to give a lot of examples because we are currently in a pandemic era, and Ebola, the epidemic that we encountered in 2014 to 16, excuse me, is one of the first epidemic to be tracked in real time using next generation sequencing data. So for understanding multi-omics integration in infectious disease biology, we don't have to go far, but we can easily take the Ebola pandemic as a case study to understand how different strains of Ebola are characterized and how different outbreaks of Ebola are actually contained by analyzing the next generation sequencing data, and how we are repeating the same for SARS-CoV data. All of your variants of concern and variants of least concern and double mutant, triple mutant of these variations of these Delta and Omicron variants are actually employing next generation sequencing data for their characterization. So as a result, as we know that the omics data is is now regularly used to study viral genomes, is used to study DNA variations, is used to study pathogenic gene expression or not pathogenic gene expression, host gene expression data, and host protein abundance, and the composition of different properties of like microbial community in the metagenomics data. So all of these different types of data that we have need to be effectively analyzed and need to be mined for different patterns to understand how hosts respond to the pathogenic invasion, and how generally pathogen employs different parts of host protein machinery for its own purpose. So in the end of all these, you will understand and you will develop a deep understanding in the processing and analysis methods that are used to integrate genomic, transcriptomic, and metagenomic data, so that you can mine in the we don't have CRISPR also, but if you can find CRISPR data that will complement omics data, nothing stops you from using that, Shiva. So yeah, we give you methods to study relationship based on for example local and multiple sequence comparison using phylogenetic analysis, and phylogenetic analysis will give you a new world into the reconstruction of evolutionary patterns for example for viruses and for different pathogens. We also give you different for example chemical and structural variations that is observed in the sequence, amino acid sequence, that are mapped onto the protein from, for example, in the surface protein of SARS, on the surface protein of Ebola, or in the genes of Mycobacterium tuberculosis, and etc. And we also give you ways to understand the immunology, immune response of the host by studying their transcriptomics data, transcriptomics landscape for a pathogenic invasion. So with that, I can take some questions that you are busy with, or you are typing with. And then if you have no further questions, I will pass the stage to Dr. Mohit, who is going to talk to you about research fellowship.